Hello, everyone. It's for me a great pleasure to virtually welcome Dr. Cédric Raoul. Cédric Raoul is Research Director of the National Institute for Health and Medical Research in Montpellier, France. Dr. Raoul started his work on amyotrophic lateral sclerosis during his PhD in Marseille in the Professor Chris Anderson lab. He then did his postdoc with Professor Patrick Abicher in Lausanne, Switzerland, where he focused on gene therapy for ALS and established the proof of principle of viral mediating silencing of SOD1 as a therapeutic approach to ALS. Since 2006, Raoul's team has proposed to explore the contribution of non-neuronal cells to motoneuron vulnerability to design innovative therapeutic intervention. Toward this aim, he built up a multidisciplinary team with a big expertise on standard molecular and cellular biology of motoneuron, electrophysiology, preclinical aspects, and clinical genetics of ALS. Welcome, Cédric.
Welcome. Good morning, everyone. I don't know if you hear me properly.
Yes.
Okay, good. So thank you, Anaïs, for this, invitation and introduction. And thanks to all of you for joining us to this very exciting meeting. I don't know if you can see my presentation. Everything is clear, or do you see my slide?
No.
We
cannot.
Wait. Okay, it's going to work.
Okay, perfect.
Good, okay.
Thank you.
Okay, thanks to you. Okay, so basically I'm going to talk about mechanisms that leads to motoneuron vulnerability in motoneuron disease, the most common motoneuron disease, which is amyotrophic lateral sclerosis. So as you know, motoneurons represent the final integrative step in all voluntary movement and commands. So you know that motoneurons are located in the motor cortex, in the layer 5 of the premotor and motor cortex, and they are glutamatergic neurons. They send their projection, so either directly to lower motor neurons or through rebospinal tract. Here it's not represented, but you have a direct connection between motor neurons, upper motor neurons, and lower motor neurons that you find in the brainstem, and also all along the spinal cord. And these lower motor neurons contact directly striated muscles and control in a coordinated manner muscle contraction. So when things goes wrong, it can lead to very debilitating disorders. And the most common adult motor neuron disease is amyotrophic lateral sclerosis. So ALS is characterized by the selective and progressive degeneration of upper and lower motor neurons. It's important that both groups of motor neurons are affected in the disease. So prototypic ALS starts in a focal, in very distinct place, group of muscles, and is going to evolve very rapidly to a generalized paralysis, leading to death of patient mainly by respiratory failure within 3 to 5 years after onset. So, most of the cases are sporadic, as a lot of neurodegenerative disease. And, it's unknown why it starts in the family, why one person will develop ALS. And there is a lot of molecules that have been, postulated. Most of them may be considered a risk factor. And in 10% of cases, it's a familial ALS and it's inherited. Actually, more than 25 causal genes have been identified so far. The first gene which has been identified is the gene coding for the superoxide dismutase 1. It was in 1993. And it represents about 20% of familial ALS. It means roughly 2% of all cases. It's an autosomal dominant inheritance, and there is a lot of mutation which has been described so far, most of them are missense mutation, and they are scattered throughout the whole enzyme. There is no hotspot. I mean, you can find from the C terminus to the N terminus in the, catalytic site. Everywhere you can find mutation causing ALS. And quite quickly after the discovery of this mutation in humans, group have developed some transgenic mice that all express the human mutated SOD1. Most of them use endogenous SOD1 promoters. You've got ubiquitous expression of SOD1, but it leads specifically to a motor phenotype. So, and these mice have been very helpful because basically they display most of the clinical, electrophysiological, and histopathological feature of the human disease. So you've got a selective loss of motor neurons, but it's interesting to see that in patients and in mice, it's been well described for sure, better in mice. But the slow and relentless degeneration of motor neurons is also accompanied by, an activation, proliferation of glial cells. So it's mainly astrocyte and microglial cells. And it's very interesting because it can start well before the onset of symptoms. So you've got the generation of motor neurons, and a lot of things happen around motor neurons, implicating glial cells. So the first, people wanted to understand what is the contribution of different cell type in the degenerative process. And it comes from a very elegant work from Cleveland group, whereas basically they use the Krelok system to specifically excise the mutated protein in some specific cell type. So you've got, expressions, the G37R in this case, with human mutation, and does the endogenous promoter. And what they did is to basically delete SOD1 in motor neurons first. And when you have, an environment expressing SOD1 except motor neurons that do not express SOD1, you see that basically you have an effect on the early phase of the disease, suggesting that indeed the toxic action of SOD1 in motor neurons is among the first pathogenic process. They did exactly the same with the GFAP promoter, where you can delete, SOD1 mutant only in astrocyte. And in that case, what you can see is that you have an effect on the progression rate of the disease, but not on the onset of the disease, suggesting that indeed astrocyte contribute to late phase or to a progression phase of the disease. And they did also exactly the same with microglial cells. And when you deplete microglial cells from SOD1 mutant, you see that basically you have an effect on the late phase of the disease. So it's a very elegant, approach to better understand what's the role of different cell type in the environment of motor neurons in basically the course of the disease. Well, okay, we know that now that astrocyte glial cells plays a role, but what kind of factors are secreted and how these cells can mediate motor neurons death. So several years ago, we started to work on this kind of, relationship between neurons and environment. And we basically describe a pathway which can be activated by the production of nitric oxide, which is can be produced by microglial cells, astrocytes too. And nitric oxide can lead to the upregulation of Fas ligands, which is a TNF family member. And Fas ligand can now activate in a cell autonomous manner is receptor, which is Fas or CD95. And Fas is well-known immune system, which is a death receptor. And Fas in motor neurons can lead to a very complex cascade of events, which is, and this cascade is specific to vulnerable motor neurons. And it seems that the vulnerable motor neurons disease have a kind of intracellular landscape that offer to Fas a way to signal and to lead to the death. So you've got implication of P38 kinase with some transcription of nitric oxide. synthase, neuronal nitric oxide synthase that can in turn also produce nitric oxide, that just amplifying the pathway. You've got also production of CRAM4A to lead to axonal degeneration. And you've got also a link to the caspase cascade, which is a standard mitochondrial pathway with cytochrome C release, caspase reactivation, caspase 9. And what makes the difference between vulnerable and not vulnerable motor neurons is basically linked to the calcium buffering and the ER, and specifically the expression of calreticulin. So you've got also a pathway involving calcium mishandling and ER stress. So it's one aspect of the relation between astrocyte and motor neurons. But what people have done after the description of fast pathway, it's an Italian group. They basically try to down-regulate fast expression using RNA interference. And here you can see that you've got an effect on the lifespan. So it's a classical 13 or 15% increase. So meaning that indeed fast is involved in the pathogenic process, but it also shows that it's not enough. That even if you basically silence this death pathway, things will at the end or eventually occur and motor neurons will die, suggesting that other pathways are also involved. Another pathway that we have identified after the fast pathway is basically the one involving interferongamma and TNF, another TNF family member, which is called light and lymphotoxin beta receptor. We have shown that astrocytes or mutant astrocytes specifically express interferongamma, which is released and activates at the surface of motor neurons expression of light, which can in turn, in a cell autonomous manner, activate lymphotoxin beta receptor, and lymphotoxin beta receptor will activate a complex cascade involving also P38 kinase and another different cascade that what you saw with FAST involving caspase 9, not 3, 6, and cleavage of lamin A. In any case, it leads to death of motor neurons. Here you have a direct effect of interferon gamma on the activation of motor neuron specific death pathway. And as you could expect, we cross the light deficient mice with the SOD1 mice, and we don't have much effect on the onset, which is quite logical because interferon gamma is expressed very late in the disease at symptomatic stage, but you have an effect on the progression rate of the disease and the life expectancy. But as usual, it's about 13–50% increase. Again, showing the complexity of the system and that by targeting only one molecule is not sufficient. So we also continue to work on the molecular pathways that could be involved in motor neurons death. And we identified basically another one, which is involving a pro-inflammatory cytokine, which is called TWIC, well known in the immune system. And TWIC is exclusively expressed in astrocytes during the symptomatic stage of the disease. And what is interesting is that basically interferon gamma can, leads to the production of TWIC by astrocyte, and TWIC can also stimulate the proliferation and production of interleukin-6 by astrocyte. So we've got something that seems to control basically the activation status of astrocyte. As usual, we cross the TWIC knockout mice with SOD1. It was quite disappointing here because we don't have any effect. So we measure the weight evolution of the mice. We give you an idea about the muscle atrophy and allow us to determine the onset. We don't have any effect on the onset of the disease when we have depletion of TWIC. You don't have any effect on the motor function. You still have a motor decline, deterioration of motor capacity, and in terms of life expectancy, it does not influence anything. So it was very disappointing. So he said, okay, we're gonna maybe take a look more precisely at the histological level. And surprisingly, what we have shown is that when you deplete TWIC in SOD1 mice, you have a massive effect on basically the astroglioses. But more important is the effect on the microglial activity. You have a 90% decrease. it's very surprising because you don't have any effect on the motor neuron, degeneration or on the course of the disease. Suggesting that, there is a lot of possibility, but basically one attractive idea is that when you arrive too late, when your intervention occur maybe too late in the process, things are so disorganized, dysfunctional that you will not be able to reverse basically what has been committed before. So it's one aspect of the disease, and specifically if you think in terms of therapy and the cell targets you have basically to address in your therapeutical approach, you have to basically intervene in the good time window. This is one aspect of the disease. And the other aspect has been interesting also, has been revealed by another effect of light. So I remind you that light can promote the generation of motor neurons through lymphotoxin beta receptor. But at the same time, when you take motor neurons in vitro, this is a purified motor neurons that you had, recombinant light to your motor neurons, you see basically a quite important effect on axonal outgrowth and the branching complexity. So at the same time, you can trigger death, but you have also an effect on the axon outgrowth and branching. Was quite disturbing. So we said, okay, since neurons are highly polarized cells, you have very specifically for motor neurons a very long axon. We thought maybe it's the matter also of site of activation, and we're going to explore this possibility. So what we have done is to use compartmentalized culture system using microfluidic chambers, where you can basically separate the axon from the soma. So we just sit in one compartment, and the axon will cross this microchannel, and you retrieve all the axon in another compartment. And it's very convenient because you can play basically differentially on axon and on the soma. So it's what we have done with recombinant light. And what we first analyze is the deaf activation. And what is interesting here is that basically when you activate light at the somatic level, you have deaf. But when you add light in the axonal compartment, you don't have any effect on the survival. So it seems that the effect of light regarding death is basically linked to the activation of the receptor at the somatic level. And when you look at the outgrowth, it's exactly the opposite image. It means that you've got to stimulate the receptor at the axon level to have an effect on the axon outgrowth and branching complexity. If you stimulate the soma with light, you don't have any effect on the axon outgrowth. So this is in vitro, and I make a long story short, but here the idea is that depending where the receptor is activated, the response of the neurons will be completely different. And in the case of nerve injury, when you have a degeneration of the nerve, we see that basically B cells, lymphocytes, basically comes to the lesion site, they express light, and here we demonstrated that activation in vivo, activation of receptor can promote axon repair, and you have a better effect, a good effect on the recovery, on the motor recovery. But at the same time, we show in NILES that light here activates at the somatic level lymphotoxin beta receptor, but here it drives basically an apoptotic program. So it's a very important message. If you think in terms of therapy, that depending on where you activate or where you stimulate your system or silence or activate, the effect can be completely different. So it means that the route of delivery and is very critical. It's very important, as we show just before with the tweak and interferongamma. Is also the time window is also important when you think about therapeutic intervention. So this is just example of the complexity of astrocyte and the microglial cells. There is more molecules that have been identified, not much actually, but there is other pathways that have been proposed to trigger the toxicity of astrocyte toward motor neurons. But interestingly, there is also another cell type that infiltrates the spinal cord during the course of the disease. So the infiltration is mainly T cells. We don't have much B cells infiltrating the spinal cord and the brain, but it's mainly CD4 and CD8 T cells. So it was, I mean, a very nice work. And from this discovery that lymphocyte can infiltrate the spinal cord and the CNS of patient and mice, we had a very massive work from Stanley Appel lab, and they basically show that the CD4 T cells basically play a role in in promoting an anti-inflammatory response and to be neuroprotective during the process. And it's mainly due to the regulatory T cells, which are known to basically suppress immune reaction. And you've got a clear link between the severity of the disease when you compare fast progressing or slow progressing patients and the level and the number of circulating regulatory T cells, Treg. So there is definitely a protective effect of Treg and a clear correlation with the severity and progression rate of the disease. And more recently, there was a phase 1, using 3 patients. It's not a massive clinical trial, but in any case, it's very encouraging. And what they have done is to basically isolate Treg and expand them in vitro and make a transfusion, autologous transfusion of this Treg in patients. So for the moment, it's still to be well tolerated. And so story is still continues. So it's a very interesting aspect of the immunity, how we can use T cells to basically maybe provide a therapeutic effect in humans. But there was also, at the same time, that you have, infiltration of CD4, you have also an infiltration of CD8 T cells. It was a very old paper in the 90s where they basically saw on the section of human spinal cord some, at least T cells, CD8 T cells. So we said, okay, we're gonna work a bit on that, and we use, gating strategy to really clearly identify CD8 T cells. It's not ambiguous. And you see that basically at 90 cc onset, and you've got an infiltration of CD8 T cells in the spinal cord, in the CNS of SOD1 mutant mice. Why in the periphery, in the blood, you don't have, massive effect. You don't have, an increase of CD8 T cells in these mice. So we look better on the activation status of the CD8 T cells, and we show that basically using CD44, CD62L immunophenotyping, we show that it's in the CNS, you've got an infiltration of memory, effector memory T cells. So suggesting that it's not just, that there is disruption of the blood-brain barrier and they just come around and after they disappear. No, basically it's a memory T cells, effector memories have been activated in the periphery and they come specifically, basically, as you will see, in the CNS of SOD1 mice. And this effector memory T cells are basically using KLRG1 marker activated T cells. So we, the standard approach, and you've got some SOD1 mice and you cross them with mutant mice that are deficient with the gene that you want, with the target gene. So we use the CD8 alpha knockout mice, and it's a very nice tool because you can have, quite important depletion of CD8, but it does not affect the other compartment of CD4, which is quite important because you know that CD4 play a role also in the disease. So we can specifically deplete the CD8 compartment in this mice. And one more time was really disappointing because when you look at the weight evolution of the muscle atrophy and the weight of animals. You don't see basically difference between SOD1 mice having or not CD8. And we don't have any effect on the age of onset. We look at, the motor deterioration capacity, motor capacity of mice, and red and green curve, you see basically there is no difference. So you can have or not have CD8 T cells, it doesn't influence anything. And it's the same with basically the lifespan of the mice. Well, very disappointing. So we said, okay, we're going to continue. We're going to keep on analyzing at histological levels. So we basically look at the motor neurons. So we use CAT staining, cholinesterase marker. And surprisingly, this is a control in black, and you have, for sure, loss of motor neurons in green here. This is just SOD1 mice. But when you have depletion of CD8, you see basically you've got an effect on motor neuron survival. So you have more motor neurons surviving. So it was quite interesting. So suggesting that basically CD8 could directly trigger death of motor neurons. So we basically use what we love to use is in vitro systems. So we purified motor neurons, and we use co-culture of CD8 T cells from wild type or from SOD1 mice and put them in culture and just look at the surviving percentage of surviving motor neurons over the time. And here you see that when you culture, motor neurons with wild type CD8 T cells, this is in blue, you don't have any effect on survival, or it's not significant from the control. But when you put CD8 T cells expressing ILS-linked mutant, you see that you can kill motor neurons, and you have indeed an effect on the survival of motor neurons. And this seems to be motor neuron specific, because when you put SOD1 mutant CD8 T cells with cortical neurons, with hippocampal neurons, or striatal neurons, you don't have any effect. So it seems that the CD8 can specifically, or let's say selectively, trigger death of motor neurons. And this is completely dependent on the expression of SOD1 by motor neurons, because you could think that, motor neurons expressing SOD1 just present some peptide of SOD1 in the MHC1, and it's just a specificity of SOD1. But basically, no, even wild type, and wild type and SOD1 mutant motor neurons are killed with the same level. So suggesting that SOD1 mutant CD8 recognize something on motor neurons independently of SOD1. So we basically, the idea here was to demonstrate that there is a direct interaction. It was not a kind of bystander effect, because we know that CD8 express interferongamma and other cytokines that could indirectly trigger death of motor neurons. So we said, okay, we're going to directly evaluate if there is a contact and interaction between CD8 T cells and motor neurons. For that, we use atomic force microscopy. So it's a single cell force spectroscopy. It's a very convenient approach. So you use AFM. So the idea is to stick here your CD8 T cells to the cantilever of the AFM. This is just a glass with your motor neurons. And you just approach, this is in red. So you start from the right to the left. So you just approach here. And this is a force in piconewton. And then after you touch, so you apply a force, you see here. So you touch and you apply a force just to get a good contact between the motor neurons and the CD8. You dwell for maybe 1 second or 5 seconds. And then after you start to detach the CD8 from the motor neurons. And you see this is a blue here curve. And you see basically that the cells detach quite easily. And when you use a SOD1 mutant CD8 T cells, you see here that the curve goes very, very low. So when you calculate a surface under the curve here, just to give you an idea of the adhesion force, and you see that white type motor neuron have non-specific interaction, you have an increase in the adhesion force when you have, lymphocyte expressing SOD1 mutant. So we use an MHC1, a blocking antibody that blocks the interaction between the TCR and the MHC1 complex, and you see that basically you can abrogate the interaction between CD8 T cells and motor neurons. So to make it short, we also describe the pathway that leads to death of motor neurons, and use exactly the same system which is classically used by cytotoxic CD8 T cells. It can be either fast ligand activation, because the activated CD8 T cells after commitment of the TCR basically express fast ligand, and fast ligand can trigger fast at the surface of motor neurons, leading to the death pathway using the classical cascade. But we also know that CD8 T cells use a granzyme perforin system, which perforin make holes in the surface, in the membrane of motor neurons, and granzyme can directly cleave caspase and trigger death of motor neurons. It's what happens when CD8 T cells recognize motor neurons. So we have also, I mean, this is not really necessary, but we have also shown using analysis of the TCR repertoire of T cells infiltrating basically the CNS of SOD1 mice, that you have indeed oligoclonal and monoclonal response of CD8, and it's mainly the TRBV15. It's not very important, but what here clearly shows that you've got autoreactive T cells, activated autoreactive T cells that infiltrate the spinal cord, and it's some specific clone expressing specific TCR that goes into the spinal cord and trigger death of motor neurons. But now the question is, what kind of peptide is expressed on the surface of motor neurons? For us, it's very complicated to know. It's not very simple. But the other aspect is that basically we have a good effect on the survival of motor neurons, but you don't have effect on the motor capacity. So we said, okay, it's very surprising that CD8 T cells kill motor neurons, but you don't have any effect on the motor function. So we go back to a very old paper, and there was an interesting paper basically showing that the neurons that become electrically silent express MHC1 of the surface, and this is potentiated by interferon gamma. So we said, okay, this is quite interesting. So we look in our model whether MHC1 was expressed or overexpressed, and indeed, at symptomatic stage, when you look at the expression of the heavy chain of MHC or the light chain with beta-2 microglobulin, you see that there is an upregulation of the MHC1 complex in the spinal cord of SOD1 mice. And we did confirm using in vitro system that when you stimulate with interferon gamma, you've got also an increased expression of MHC1 of the surface of motor neurons. And this is quite, logical with the expression of interferon gamma by astrocytes. We know that we've shown you that astrocyte can produce interferon gamma, and also CD8 T cells express interferon gamma. So this is just an intracellular staining of interferon gamma in CD8 T cells infiltrating the CNS of SOD1 mice, and you see basically there is a big expression of interferon gamma. So you've got basically everything here through astrocyte or through infiltrating T cells, interferon gamma that can stimulate expression of MHC1. But what about the electrical activity? It's known that there is problem of excitability in ALS, and it's a very nice work from Daniel Zicnicki group. They show that basically there is a lot of electrophysiological function where motor neurons become hypoexcitable during the neurodegenerative disease. So it was a good link for us to explore, and to explore whether basically CD8 T cells could kill only silent or electrically, deficient motor neurons. And we go back again to in vitro system, and we basically challenge motor neurons with CD8 T cells, mutant CD8 T cells, at different time of culture, because it's very interesting in motor neurons. When you take motor neurons after 3 days, you see that you don't have much, spontaneous activity here. This is a loose patch recording, and when even you inject currents, you don't have a massive response. Sometimes you have spike, but it's not very convincing. You have to wait at least 7 days before being able to elicit, a train of action potentials. This is very nice here, it's just increasing step of current, and you see that you have a lot of action potential, and you have also here spontaneous activity. So you've got here a way to evaluate whether CD8 T cell could preferentially kill silent or electrically active motor neurons. And basically, it's very interesting because when you put CD8 T cells on silent motor neurons, it's what we are doing since the beginning, you have killing effect. Basically, you kill motor neurons. But when motor neurons become electrically active, basically, you don't have any killing effect, suggesting indeed that the electrical activity is also something important for the initiation or recognition of motor neurons by CD8 T cells. So the idea here is that when you have a loss of repetitive firing or when you have dysfunction of excitability, basically we can have an upregulation of MHC1. We know also that when you have a loss of repetitive firing, you can have anti-activity, specific antigen can be released or produced. And we can basically imagine that this antigen, which is related to activity, can be presented by the draining lymph node, and through a system called cross-presentation, activate CD8 T cells in the periphery, and then activated T cells goes into the spinal cord to trigger death of the silent motor neurons. This is basically the idea we have. So just give you so I'm going to continue. Do I have time? I don't know. I've got 5 minutes. Okay. So we're focused on, glial cells, microglial cells, and non-neuronal cells. But we recently focused on neuronal cells because you know that I was talking about electrical activity, but what is really important in the electrical activity is interneurons. So basically, we were initially involved in the chloride conductance, chloride current, and we were basically screening for the channels that would be responsible of the chloride conductance elicited by calcium. So we use, don't go into the details, but we use a protocol for tail current, for chloride analysis, and we targeted a family of genes called TMEM16, which are calcium activated chloride channels, and we identified in motor neurons TMEM16F as a main channel responsible for the chloride conductance after calcium release. And this is a TMEM knockout mice, as you see a massive effect on the chloride conductance. So it was interesting because TMEM16F is found, when this is just a section of wild type motor neurons, is found basically in this small dot, which are choline acetyltransferase. It's what we call C-button. It's a cholinergic input on motor neurons. So you know that motor neurons which are cholinergic, this is not great news, but you have got also, some interneurons which are cholinergic. It's a more modulatory interneurons. And we found that TMEM16F is a new component, actually, of the C-button as expressed by motor neurons at the postsynaptic site. And so to make it short, we basically show that TMEM16F is regulating the recruitment threshold of motor neurons. It means that we lose TMEM16F. You see that basically you increase the threshold for, triggering a spike, an action potential. And we did that using spinal cord preparation, spinal cord culture. And you see that you can basically have you can identify the slow, fatigue resistant or fast fatigable class of motor neurons. And the most, vulnerable, basically motor neurons population is a fast fatigable motor neuron population in ALS. And the effect of TMEM16F on the recruitment threshold is basically on this specific, selectively on this population, which is the most vulnerable to ALS. So again, we took the TMEM16F deficient mice, and we crossed them with SOD1 mice. And we have here a very interesting phenotype. We analyze, as usual, the onset and the probability of survival. And we have got an effect on the onset, but only in male, but not in female. And the lifespan is exactly the same. We've got an effect on the basically male, but not in female mice. And when you look at the motor function, you see also that you got a very good effect. Yeah, it's a very good effect, but after the slope is very, very, very hard. But good effect on the motor function on male, but not in female mice. So we don't know yet. We explore for sure all the hormonal link, how the hormone can modulate, electrical activity and susceptibility, but we still don't know why it's male and not female. But what is interesting here is that basically you see that interneurons play, it's a modulatory interneurons, play a massive effect on the motor vulnerability in the disease. And it's just to illustrate the complexity of the system. So you see that here, this is a review that we have written recently. It's basically just to illustrate the complexity of the system, because in ALS in patient and mice, we know that we've got degeneration of, GABAergic, glycinergic neurons. We've got also basically long distance or long range modulatory input, serotonergic neurons, and it creates a kind of very complex imbalance of, excitatory and inhibitory regulation of motor neurons control. And it's very important for motor neuron function and degeneration at the end of the day. And we also know that oligodendrocyte plays also an important role in the disease. And there is also degeneration of oligodendrocyte well before degeneration of motor neurons, it was the same for interneurons. You can have degeneration of interneurons well before degeneration of motor neurons. And it involves a lot of complex signaling that at the end disturb the function of motor neurons and leads to a complex cascade activating astrocyte microglial cells and also lymphocytes. So at the end of the day, it's very complex. So I would like to thank a lot of people in the lab and collaborators. They follow us and help us during all these years of research, and for sure the funding agency that basically without them we would not be surviving. Okay, thanks for your attention. I take any kind of question.
Okay, thank you a lot, Cédric. So there is a question. Could you elaborate your view on the role of intracellular calcium and ER stress in the death of these motoneurons?
Yes. So ER stress, I'm going to share. Where is it? It's not new that you see my screen?
Yes.
Okay, good. So it's not, completely new if I tell you that the ER stress occur very early in the process. It happens in most of the neurodegenerative disease, especially neurodegenerative disease where you have aggregate of misfolding. And that's the case for SOD1 and other, linked gene associated to ALS, TDP43, for instance. But basically, ER stress leads very early in the process. It's a work from Pico Caroni lab where they show that the susceptibility or motor neuron susceptibility is basically linked to an early activation of ER stress and the ubiquitin pathway. And here what we have shown also is that activation of FAS leads to the downregulation of calreticulin, which play a major role in calcium handling and ER stress. And indeed, after you've got a release of calcium, and calcium have a lot of function. For sure, one of the well-known function of calcium is very historical. It's excitotoxicity, but here it's not this kind of death we have. But in motor neurons, and specifically in ALS vulnerable motor neurons, you have a complex cascade following calcium activation, and specifically, as you saw recently, as I shown you, activation of chloride channels. So the link between chloride channels and cell death pathway for the moment, we still don't know. But yeah, I don't know to which level I can go, but in our case, the downregulation of calreticulin is a signature of vulnerable motor neurons.
Okay, thank you.
We have another question. It sounds treating patients before the onset of symptoms is important. Are there any biomarkers which detects pre-symptomatic ALS patients?
The point is to find, it's important. It's indeed an important point that the earliest you arrive in therapy, the better will be. There's not much doubt about that. Except some, for sure, I will tell you the genetic is the best biomarker that you can have. If you have a family, a known family, for sure you can identify it. People who are carrier of genetic mutation, knowing that a lot of mutations, specifically SOD1, has a high penetrance. So basically you can identify on the familial tree, using familial tree, basically what kind of patient will potentially and eventually basically develop the disease. For biomarkers, you don't have much, or specific biomarker, you don't have much. The one which is basically the most advanced is the neurofilament. It's basically one which is quite good, but it's not, again, it's not really specific to motor neuron disease. But there is no, to my knowledge, one biomarker or combination of biomarkers that will ensure you, okay, we are in the early phase of the disease. You've got good biomarker, reliable biomarker. Neurofilament is a reliable biomarker. But to provide you something that will, to the clinician, help you to say, yes, you're going to develop the disease in 1-2 years, it's quite tough to find, to isolate. And there is a lot of work in using classical protein or metabolomics and so on, try to identify some combination of marker. But there is the complexity also, it's that basically patient goes to see the neurologist when they are ready, when the disease is already well advanced. You know that there is a period of about 12 or 14 months where the patient basically just doesn't know what they have, and they can wait before the first weakness or the first sign before this time. And when they start to see the neurologist that will quite rapidly diagnose ALS, there is a period of 12 to 14 months, which is huge, where the patient doesn't know what he has. And it goes from service to another one. So there is also this challenge. Basically, if you want to intervene early, is to reduce this time. And so it's quite complicated and it's challenging, but I think we're going to find eventually a solution for that, but it takes time.
Thank you for your answer.
Yeah, there is another question. UPR is a protective pathway also in neurons. Do you think there is solid evidence that chronic UPR that leads to ER stress is actually detrimental to neurons?
Yeah, it's quite, in vitro, yeah, for sure. In vitro, we've got a lot of evidence. In vivo, it's more complicated, or it has not been well addressed using drugs, tunicamicin, salubrinal, or these kind of things. Or there was also XBP1 deficient mice that show some, they can suggest that indeed the UPR is indeed involved. Yeah, genetic approach with sensor can help actually, but solid, I don't know, solid, to which point is it in terms of patient? I don't have much. Yeah, genetic approach, yeah, they show, and some pharmacological, there is not much pharmacological approach, and genetic approach demonstrate that the UPR, chronical, basically chronic activity, chronic response can be detrimental. Yeah, there is some, at least in mice, there is some evidence that the system is bad when it's done always on. Yeah, don't know what to say more.
Okay, thank you.
Maybe a more general question now. It was very difficult from the literature to find studies that would then investigate the environmental risk factors to explain the sporadic form of ALS. So would you have any ideas about why these factors are so hard to pinpoint?
But this is basically the definition of environment. So you can have, since, motor neuron is an adult disease, so it means that you are exposed. Oh, there is another question, is when really ALS develop, if it's a developmental disease or if it's just only adult disease. But during all your life, you are exposed to so many different factors. How could you identify one? For sure, there was some good, the cluster of Guam is very interesting with the BMA, neurotoxin, and also pesticides. There was also all this stuff about Gulf War, Army. It's, it's unknown. So the easiest way to answer is basically a combination of the genetic landscape or susceptibility to a specific environment. So it's I think most of the sporadic cases are basically associated to a combination of really environmental factors, exposition to heavy metal, if you have a bad, life quality, smoking, alcohol, drugs, and so on, combine basically to, susceptibility gene. There is more, if you take, susceptibility gene and causative gene, you have more than 50 genes associated to ALS. So it's a very complex interaction, and but it's really, really complicated. And if it was simple, everyone will know why Alzheimer is mainly sporadic and all this stuff. So, but let's say it's a combination of both system and genetic and environment. So environment means it could be anything. So potentially, depending on where you work, what kind of diet you have, your lifestyle, and so on. So. Unfortunately, I cannot answer clearly to this question.
Okay, thank you. Claudio Rivera has a question. Changes in intracellular chloride concentration is involved in volume regulation, and regulatory volume changes can be involved in apoptosis. Do you think the effect of calcium-sensitive chloride channels may be related to this process?
This is, thank you, Claudio, for this question. This is indeed a very good question. We don't know. We don't have the feeling that the volume changed drastically. We didn't look at actually precisely. We didn't explore these aspects of indeed volume control. We didn't do that, and or maybe. Yeah, it's an important aspect, especially with linked with death. So unfortunately, we didn't do any experiments on that. I don't have any. I don't have anything to tell you about that actually. Yeah, I'm quite dry.
Well, thank you. Maybe we have time for one last question, which would be, with the scope of RNA therapeutics rapidly expanding and the genetic basis of ALS continuing to be uncovered, which future therapeutic developments seems more promising to you?
Gene therapy. That's right. I mean, look at the effect of, what we have with spinal muscular atrophy is just amazing. I mean, it changed the life of the patient completely. It's basically changed because they saved the life. So gene therapy is now a very it's a realistic strategy. It's no more something that we could potentially do. No, it's already in the clinics, and look at the effect that you have by a gene replacement strategy. SMA, or when you try to restore gene function with SMN, you see that it's really impressive. So gene therapy is something that we should push forward. And because now there are more and more vectors, more efficient, safer, and you can produce big scale of very safe vector and efficient vector. You can now have vector with a very good tropisms when you can target specifically, neuronal cells. It's more and more attractive and more promising, and I'm convinced and gene therapy will explode. And for sure, if you're, a bit cold with gene therapy, you still have the approach which is developed in ALS with antisense delivery, intrathecal administration of antisense, which is also approach with SMA. So no, it's not just a sci-fi. It's something that happens, and there is something going on with gene therapy and genetic approach or gene transfer approach with antisense or siRNA. It's for me, it's something very important. But again, it's very restricted because you have to. It's restricted to the genetic cause. It's easy when you have basically identified the genetic cause. It's straightforward approach. But again, it represents a small proportion of patients. Look at C9 or SOD. You don't. It's basically C9 is different. There is a lot of patients, but with C9 mutation. But even it's not. It's very good for people who can be treated for sure. But there is still a lot of patients that we have to address. So, but gene transfer is a very good approach, I would say.
Okay, thank you a lot, Cédric, and thank you all for your question.
Thanks to you. Thanks for.
Now we have a little 5-minute break, and then we are back for the panel discussion about brain disorders.
Okay.